This is the last part of my talk, in which I'm going to discuss two additional predictive modeling approaches applied to pre-manifest HD. And first, I'm going to start with an analysis that incorporated trajectories of multiple subcortical structures for the analysis of pre-manifest HD. The most salient marker or hallmark of pre-manifest HD is the atrophy present on one subcortical structure called the striatum, which I believe I mentioned earlier, referring to the caudate nucleus, a substructure of the striatum. In any case, the atrophy in the striatum is pretty evident in pre-manifest HD. However, other subcortical structures also undergo significant atrophy, but not to the same extent as the striatum. Still, considering that multiple subcortical structures have early atrophy prior to disease manifestation, it makes sense to jointly characterize these subcortical regions, as opposed to looking at each structure separately. So the point of this analysis was, can we identify a composite marker of subcortical volume change associated to the disease that can be robustly detected across several cohorts? And second, can we find which region interactions are the ones that drive or can inform us about pre-manifest HD pathology? For this purpose, we extended our data set, including not only data from TrackHD, but also from another big study called PredictHD and a smaller one, a single site study, nevertheless still having around 60 subjects, called IMageHD. Overall, we have more than 500 subjects for this analysis. So it is a significantly high sample size, the one used in this study. And the analysis is as follows. We have brain scans from a given individual across multiple visits. The idea is to use that information to do a segmentation of the subcortical structures. With that information, we can get volume estimates from each of these structures. And based on that information, we can look at the trajectories or the rate of change, the linear rate of change of each of them. And once this rate of change is known, we can integrate these values, plug these values into a classifier, and see if we can detect changes in subcortical structures that are characteristic to either pre-manifest HD or healthy control. So we are actually characterizing differential trajectories between healthy controls and pre-manifest HD individuals. And the classifier of choice was logistic regression, again. And in order to look at region interactions, we used partial correlations. But first, let's look at how well the model performed across these studies. Let me tell you first that the first value that you see in each of these entries in the table, this first value is the prediction, not the prediction accuracy, but the AUC, a measure of performance, the area under the ROC curve achieved by the classifier when analyzing all or incorporating all of the subcortical structures. And in parentheses, you can see the value attained by looking at just at the striatum. That being said, there are two main findings or two important findings from this table. First, when we did a validation by leaving out a whole study, we saw that we could find an AUC, a significant AUC of 81% to discriminate pre-HD individuals and healthy controls across cohorts. So the answer to the question of can we reliably detect multivariate change, volumetric change in the subcortical structure, the answer is yes, we can reliably detect it. In addition to that, we evaluated the performance of this model across different studies. And what we saw is that for TrackHD, we see that the classification jumped from 86% to 91%, 86% being attained just with the striatum, and 91% being the value obtained by incorporating all subcortical structures. So this is like there's a statistically significant difference between these two values, which in turn tells us that the performance is improved by incorporating several subcortical structures. So we know that there is an advantage to using several subcortical regions for this analysis. As I said before, the idea of these approaches is to gain an intuition as of what's happening in the brain, and that's the reason why we perform the following analysis. So we did 3 analyses. First, we looked at the differences between healthy controls and pre-manifest HD for each structure separately. Then we looked at the classification weights obtained by the logistic regression classifier, which in the end is a proxy of how well a region is doing when analyzed in conjunction with the other structures. And then we did an analysis to see which associations, pairwise associations, were informative for this analysis. So first things first, when analyzing each region separately, we can see that obviously the regions of the striatum, again, the caudate nucleus and the putamen, have a significantly different. The rate of change in pre-manifest HD is significantly higher than the one observed for healthy controls. So we see a significant reduction in the caudate, in the striatum in general, in pre-manifest HD compared to healthy controls. Now, a region that is interesting in terms of the degree of atrophy is the thalamus, which again, it is significant. There's a significant difference between healthy controls and pre-manifest HD, but it's not as such a big difference when compared to the striatum. But when we analyze the multivariate weights assigned by the classifier, we can see that the thalamus is rather conspicuous. In fact, it is the region that has the highest weight, the highest associated weight when analyzing the difference between healthy controls and pre-manifest HD. So this tells us that the thalamus, when analyzed in conjunction with other structure, it has a significant effect in the classification. So we know obviously that the striatum is important because it has like a significant amount of atrophy in pre-manifest HD, and we get a hint that the thalamus is probably an informative structure. But how so? So it is informative when analyzed with the other structures. Can we pinpoint which association is the most informative? And this, it is here where the partial correlation analysis can tell us which associations are informative for the classification task at hand. And what you see over here is the directionality of the associations between the rate of change in healthy controls, or here, actually you can see here, the partial correlations between the rate of change of volume change between regions in healthy controls, and the same on pre-manifest HD here. And here, the big plot that you see here to the left is the difference, the directionality. Blue meaning that there's a reduction in pre-manifest HD, and red meaning that there's an increase in the partial correlation between two structures in pre-manifest HD. And what we see here is, well, first of all, that the caudate and the putamen, two regions that are part of the striatum, have a stronger association, an increased association in pre-manifest HD. Let's say it happens with the caudate and the pallidum. And when talking about the thalamus, we can see that there's a reduced association between the thalamus and the caudate, which tells us, or at least we interpret this as a finding that tells us that there's a reduced association between these two structures, potentially suggesting that there's a decoupling mechanism between them. So while they undergo significant atrophy, their trajectories are not that correlated. And it is by virtue of this association that the thalamus has with the caudate, which undergoes significant atrophy, that it becomes in turn informative of the detection of pre-manifestation. So again, this gives us an intuition of the atrophy processes happening in the brain, and looking at these effects in an integrated way as opposed to an isolated way. Finally, we did an analysis along with Shreyas Farnabis, which probably by now has already given his presentation to the attendees of the DIPY workshop. And the idea here was to integrate multiple diffusion microstructure measures to analyze abnormalities in the structural connectivity of pre-manifest HD. So the idea here is that the diffusion model yields different kinds of information. Probably you also looked at information regarding the different diffusion models analyzed by DIPY. So the idea here is, can we integrate these multiple complementary sources and get a composite measure of changes in the connectivity of the brain? To do so, we looked at information from the TrackHD study from 68 healthy controls and 72 pre-HD individuals. As you could see from the previous slide, we looked at 6 spatial maps that were derived from 3 microstructure models. And we analyzed 2 different approaches, macro approaches. So first, we looked at, actually you could say there are 3 actually. We could look at each microstructure or each spatial map being analyzed separately. It will be just a single modality. I'm going to use the term modality here. A single modality being analyzed differently or separately, independently. Then we did early fusion, which is basically concatenating the information from each of these modalities together as a big vector, big chunk of vectors, and plugging in that into a classifier, an SVM in this case. And the final approach, the one we were interested in, was to integrate these different modalities in a more intelligent way in which the model actually learns the associations or the integration between these different modalities. And we used, in order to do this, we analyzed 3 different approaches. First of all, we looked at multiple kernel learning, which in the context of this analysis can be thought of as an extension of supervector machines. And why is it an extension? Supervector machines basically has a single kernel for the entire data that is provided to it. Whereas in multiple kernel learning, you have 1 kernel per modality. As you can see depicted here, the modalities again would be the spatial maps derived from the microstructure models. And the model, what it does, it learns a linear combination of this kernel. And this combination, optimal combination of kernels, is in turn provided to a classifier. The second approach being analyzed here was multi-view boosting, which, again, talking about extensions, could be seen as the extension of AdaBoost to multimodal learning. And AdaBoost, for those of you who are not aware of what I'm talking about, is basically an ensemble learning model that basically learns multiple adjustments in the data in order to adjust for potential errors. So basically it polishes the errors of a given model by basically stacking together different models, making an aggregation of models, if you wish. So the extension here is that basically this does sort of an AdaBoost is applied to each view, and there are adjustments done in a modality specific basis. So basically we have like a stack of different base learners per modality, and in the end, these learners are put together across modalities. And finally, we also tried an approach using a fully connected neural network in order to combine the information provided by each of these modalities. What we found was that, as you can see first by looking at each modality independently, we found that mean diffusivity and free water diffusion provided the best performance, achieving 85% AUC. So this was the baseline that we needed to improve upon in order to be able to conclude that our multimodal integration approaches were useful for the purpose of differentiating between healthy controls and pre-manifest stage individuals. And as you can see over here, multiple kernel learning was able to achieve a 90% accuracy, or excuse me, AUC in this case. So it went from 85% AUC to 90% AUC, obtaining a significant improvement over the baseline of 85%. And again, trying to understand what was driving this improvement in terms of performance, we could see that fractional anisotropy, mean diffusivity, and free water DTI were the most informative, or the modalities that could contributed the most to the characterization of white matter microstructure in PhD. So to conclude, first of all, I was able to demonstrate the value of brain imaging for prospective estimation of clinical outcome, both in terms of motor impairment and cognitive decline. Also, we detected predictive patterns of subcortical atrophy by incorporating multiple subcortical structures. And in addition to that, we also found informative associations between these structures that provide an intuition of disease dynamics. And finally, we proposed a framework, a multimodal framework, to integrate complementary measures of diffusion microstructure to characterize pre-manifest stage. I want to thank you very much to the organizers of the DIPY workshop, and also to the attendees of this fantastic online workshop. Thank you very much for your attention.